Welcome to our session, New Developments in Correcting the Basic Defect. My name is Meg Sate, and I'm moderating this session with Professor Isabelle Sermet-Gaudelou. The objectives of our session are 1. To describe the different ways in which antisense oligonucleotides can be utilized to inhibit nonsense-mediated decay of truncated CFTR protein produced by premature translation termination. 2. Identify the benefit of delivering CFTR gene therapy through a lentivirus vector in the neonatal immune-privileged stage to achieve a greater number of transgene-expressing subjects via neonatal airway versus aerosolized mRNA replacement therapy that is CFTR mutation agnostic. And 3. Explain the importance of having a reliable airway epithelium system capable of helping researchers and clinicians understand specific CFTR variants as well as individual subject response to modulators in order to personalize and tailor therapeutic options. Here are the abstracts that will be presented. Thank you, and we hope you enjoy this session.
Today I'm going to share about our work entitled CFTR mRNAs with nonsense codons are degraded by the SMOG6-mediated endonucleotic decay pathway. Here are my disclosures and my acknowledgments. There's many people here at Ionis and at the Cystic Fibrosis Foundation that need to be acknowledged for their many years of hard work related to this project. We're all well aware that cystic fibrosis is a devastating disease. It's caused by loss-of-function mutations in the CFTR gene. Now, about 10% of all CF patients carry nonsense mutations in at least one copy of their CFTR gene, which results in the generation of a premature termination codon, or a PTC. Now, PTCs activate the nonsense-mediated decay pathway, or NMD pathway, which will result in reduced CFTR mRNA levels and very little truncated CFTR protein production. Now, it's important to recognize that the NMD pathway is not only an RNA surveillance pathway, but a post-transcriptional regulatory pathway governing approximately 10% of the total transcriptome. NMD pathway is a branch pathway, with each branch regulating subsets of endogenous RNA substrates. Now, this branch nature of the NMD pathway provides opportunity for specificity for targeted intervention for CF. So here's our therapeutic strategy for targeting nonsense mutations in the CF disease context. Our strategy is threefold. First, promote premature termination codon read-through, so as to hopefully produce full-length CFTR protein. 2, inhibit NMD pathway, so as to upregulate or stabilize the CFTR mRNAs containing the PTCs, which will hopefully produce increased partially functional CFTR protein, as well as full-length CFTR protein from basal translational read-through. And then 3, combining NMD inhibition with translational read-through. We'll evaluate this therapeutic strategy using antisense oligonucleotides as therapeutic agents. Antisense oligonucleotides, or ASOs, is a validated drug discovery platform. They're single-stranded synthetic DNA molecules about 18 to 25 nucleotides in length and interact with RNA by Watson-Crick hybridization. There are several mechanisms of antisense oligonucleotides. The one in this project we're focusing on is RNA degradation mediated by the RNase enzyme. So there's several gaps in our understanding currently related to our therapeutic strategy. First, it's unknown which branch of the NMD pathway regulates CFTR mRNAs containing nonsense codons. 2, it's unknown if the position of the nonsense codon in the CFTR mRNA will affect NMD pathway regulation. And finally, it's unknown if branch-specific NMD pathway inhibition with translational read-through inducing drugs can alleviate CF disease phenotypes. So we've generated a workflow to determine the regulation of CFTR mRNAs by the nonsense-mediated decay pathway. So we employed the Cystic Fibrosis Foundation's 16-HBE genome-edited cell model system, with each cell line containing CFTR nonsense mutations either early, middle, or late in the transcript. Then we used antisense oligonucleotides to reduce the expression of NMD factors, such as core NMD factors or branch-specific NMD factors. And then we evaluate CFTR mRNA expression, protein expression, or CFTR function by the Using chamber assay. And then these data we used to determine the NMD branch responsible for CFTR mRNA degradation. So as previously mentioned, we employed the CFF 16-HBE genome-edited in vitro cell model system to study the CFTR nonsense mutations. These cells were generated at the Cystic Fibrosis Foundation and provided to us for use. We employed 4 16HB cell lines, each containing distinct CFTR nonsense mutations, Y122X, G542X, R1162X, and W1282X. Here, we confirmed that each of these cell lines harboring CFTR nonsense mutations had reduced CFTR mRNA levels as well as CFTR protein levels. And CFTR function evaluation by Using chamber confirmed very low basal CFTR function in these cells when compared to parental cells with wild-type CFTR. So next we wanted to observe if we could restore CFTR mRNA levels in 16HB cells harboring CFTR nonsense mutations. So first we evaluated our ASOs targeting NMD factors in 16HB parental cells. We found that ASOs targeting UPF1, SMOG1, SMOG8, and SMOG9 had very good target reduction in a dose-responsive manner. When we evaluated endogenous NMD substrates following ASO treatment, we see a dose-responsive increase or upregulation of endogenous NMD substrates, indicating effective NMD pathway inhibition. Of note, for all other NMD factors that we evaluated in this study, we see very similar degrees of NMD factor target reduction following ASO treatment and anticipated NMD pathway inhibition. So when we evaluated these ASOs targeting UPF1, SMOG1, SMOG8, and SMOG9 in 16HB cells harboring CFTR nonsense mutations, we confirmed that CFTR mRNAs containing these nonsense codons are bona fide NMD substrates, as UPF1 and SMOG1 ASOs in 16HB cells harboring Y122X, G542X, R1162X, or W1282X restore CFTR mRNA levels to levels of wild-type CFTR in parental cells, as indicated in this orange dotted line. However, ASOs targeting SMOG1 regulatory factors, SMOG8 and SMOG9, alone or in combination, did not upregulate CFTR mRNA in any evaluated 16HBE cell line. Next, we evaluated ASOs targeting NMD factors involved in the recognition and tagging step of the NMD pathway. We found several interesting observations in these groups of experiments. First, we found ASOs targeting UPF2 upregulated CFTR mRNAs containing G542X, R1162X, and W1282X nonsense codons. However, ASOs targeting UPF3A or UPF3B alone did not upregulate these CFTR mRNAs. As previous reports have shown that UPF3A and UPF3B can be functionally redundant in regulating endogenous NMD substrates, we combined ASOs targeting UPF3A and UPF3B. And we found that combination treatment upregulated CFTR mRNAs containing G542X, R1162X, and W1282X CFTR mRNAs. However, none of these observations were observed in the 16HBE cells harboring the Y122X nonsense mutation, as you can see here. This could be due to the fact that there's several alternative translation start sites directly downstream of the Y122X PTC. So translation reinitiation events directly downstream of Y122X could remove the exon junction complexes, of which UPF2 and UPF3 can closely associate, and thereby stabilize the transcript. To provide further evidence of this potential explanation, we treated 16HB cells with Y122X and W1282X nonsense mutations with ASOs targeting the exon junction complex core factor EIF4A3. And interestingly, we found that 16HB cells with the Y122X nonsense mutation did not have stabilized CFTR mRNA levels after treatment with this ASO. However, we did see upregulation following EIF4A3 ASO treatment in the 16HB cells harboring the W1282X nonsense mutation. Finally, we evaluated ASOs involved in the decay step. Interestingly, we found that ASOs targeting SMOG6 upregulated all evaluated CFTR mRNAs in the 16HBE cells. And the upregulation was comparable to wild-type CFTR mRNA levels in parental cells. However, we did not see any upregulation of CFTR mRNAs following ASO treatment targeting SMOG5, SMOG7 alone, or in combination. These results indicate that CFTR mRNAs with nonsense codons are degraded by the SMOG6-mediated endonucleotic decay pathway rather than the SMOG5-7-mediated exonucleotic decay pathway. To confirm these results, we obtained primary patient human bronchial epithelial cells from the Cystic Fibrosis Foundation, which were homozygous for the W14A2X nonsense mutation. HBEs were differentiated on transwell inserts for more than 28 days at ALI before ASO treatment for 6 days. Here we confirm that SMOG6 ASO treatment upregulated W12A2X CFTR mRNAs, as where SMOG5, SMOG7, or SMOG5-7 in combination ASO treatment did not stabilize the transcripts. In summary, we found all evaluated CFTR mRNAs with nonsense codons were regulated by SMOG1, UPF1, and SMOG6. However, CFTR mRNAs containing G542X, R1162X, and W12A2X, but not Y122X, were regulated by UPF2 and UPF3A and B. This brings us to ask, could SMOG6 ASO be a pan-NMD inhibitor for CFTR mRNAs with nonsense codons? And could SMOG6 inhibition improve translational read-through therapy and increase CFTR protein expression and function? So we evaluated combinations of ASOs targeting SMOG6 with translational read-through agent G418. We first evaluated this combination in 16 HBE cells harboring the Y122X nonsense mutation. As anticipated, we found that SMOG1 and SMOG6 ASO alone did not upregulate Y122X CFTR protein, as seen here in the image, and quantified. G418 treatment alone moderately upregulated CFTR protein. However, combinations of SMOG1 ASO and SMOG6 ASO with G418 further upregulated Y122X protein expression and moderately upregulated CFTR function as assessed by the Using chamber assay. We next assessed the same treatment combinations, although this time in 16 HBE cells with W12A2X CFTR nonsense mutations. We found ASOs targeting SMOG1 or SMOG6 was sufficient to upregulate a C-truncated CFTR protein product, as seen here and quantified here. The upregulation of the C-truncated W12A2X protein product was sufficient to increase CFTR function to levels comparable to G418 treatment alone, as assessed by Using chamber assay. Interestingly, G418 treatment alone also increased the expression of this C-truncated W12A2X protein product. Now, when we combine SMOG1 and SMOG6 ASO with G418, we see only a moderate or a trend of increase of this C-truncated protein product. However, we see a far more substantial increase for CFTR function as assessed by the Using chamber assay for this combination. This could be due to increases of full-length CFTR protein in the 16 HBE cells that could not be detected by Western blot, but the far more sensitive Using chamber assay could detect. Similar results were observed for 16 HBE cells harboring the G542X and R1162X nonsense mutations. Even though we're able to increase and upregulate CFTR mRNA levels to levels observed in parental cells containing the wild-type CFTR mRNAs, highly effective translational read-through agents are likely required for maximal CFTR upregulation. So we asked, can combination of ASOs targeting translation termination factor ERF1 improve the G418 promoted translational read-through of the stabilized CFTR transcripts? So we developed and evaluated ASOs targeting translation termination factor ERF1. We found very good target reduction at the mRNA level as well as protein level in 16 HBE cells. So we next evaluated combinations of ERF1 ASO and G418 with or without SMOG6 ASO. We first started our evaluation in 16 HBE cells harboring the Y122X CFTR nonsense mutation. Interestingly, we found 16 HBE cells with the Y122X mutation treated with ERF1 ASO and G418 had a further enhancement of CFTR function when compared to SMOG6 ASO and G418 treatment. Interestingly, when we combine SMOG6 ASO with ERF1 ASO and G418, we see even further enhancement of CFTR function evaluation by Using chamber assay. We evaluated the same treatment combinations, although this time in 16 HB cells with the W12A2X CFTR nonsense mutation, and found interesting results. We found that ERF1 ASO alone was sufficient to increase CFTR function to levels comparable to SMOG6 ASO and G418 treatment alone. When we combine ERF1 ASO with G418, we see an increase of CFTR functions comparable to that of SMOG6 ASO and G418 treatment alone. However, when we combine SMOG6 ASO with the RF1 ASO with G418, we see even further enhancement of CFTR function. In summary, we found all evaluated CFTR mRNAs with nonsense codons are regulated by the SMOG6-mediated endonucleotic decay pathway rather than the SMOG5-7-mediated exonucleotic decay pathway. We also found that ASOs targeting UPF2 and UPF3 proteins upregulated G542X, R1162X, and W12A2X CFTR mRNAs, but not Y122X CFTR mRNAs. We also found that inhibiting NMD in combination with G418 or ERF1 ASO further enhanced CFTR functional improvement in 16-HBE cells. I thank you for your attention. If you have any questions related to this presentation or interest in collaboration, please reach out to Lulu Huang with her email provided here. And if you'd like to apply for an Ionis pulmonary postdoc position that's available, please scan this QR code. Thank you very much.
Thank you very much for this very, very interesting presentation. So I'm Isabelle Sermet, Rodolus. I'm one of the chairmen with Megana. So I will begin with the first question. As the NMD factors, including SMAC6, also probably affect many transcripts, how do you envision using this approach as a therapy, specifically targeting CFTR nonsense mutations?
Yeah, that's a great question. That's one of the aims of this project, was to look at branch-specific NMD factors such as SMOG6 and UPF3 proteins, UPF2, because they can those branches can regulate smaller subsets of endogenous NMD substrates compared to like the core NMD factors such as UPF1 and SMOG1. So potentially targeting those NMD factors could have less effects, as the NMD pathway can regulate about 10% of the total transcriptome. And so we have looked at that a little bit in the past, where we've shown that targeting branch-specific NMD factors such as UPF3B actually affects like about 2% of the transcriptome. And SMOG6 also had reduced total effects on the total transcriptome when compared to like the core NMD factors. So that, combined with potentially like local delivery in the lung, could be an approach for this for targeting SMOG6. Yeah.
Okay, thank you very much. A second question as well. So have you tried this combo therapy? So I guess SMOG6, ERF1 and G418 in HBE rather than in the 16 HBE from the CF Foundation?
Yeah, so we have tried targeting the NMD factors in the HBE cells. We evaluated every single combination of ASO in the primary HBE cells, as we did in 16 HBE cells, and then evaluated the CFTR mRNA upregulation. And we haven't yet evaluated any of the functional improvements in the primary HBE cells. But as far as the CFTR mRNA upregulation, it was very consistent for that in the primary HBE cells harboring the W12A2X nonsense mutation, which I think was in this presentation. It was very consistent with what we saw with the 16 HBE cells.
But still, you show some differences between the 16 HBE and the HBE as well. So how do you explain this?
Well, as far as the data that I showed with the differences between the SMOG6 targeted ASOs and the SMOG5-7, those were very consistent. So we showed that the depleting SMOG6 NMD factor levels upregulated CFTR mRNA, but that wasn't observed when we targeted the exonucleotic decay factor SMOG5 and SMOG7 alone or in combination. And so that data was in support of what we observed for the 16 HBE cells. But also, like, we haven't tested all of the different nonsense mutations in primary cells as we did test in the 16 HBE cells. And the primary cells are obviously really important, but very rare, especially to get a homozygous mutation.
Oh, and you show also very interesting data for the Y122X mutation, which is a very, very interesting mutation as well. So,
Yeah.
and also because you show that the CFTR mRNA are recognized by UPF2 and UPF3, so 4542X, W1282X, but not Y122X.
Yeah,
Right?
yeah, that's right. Yeah, yeah.
And you say maybe it's due to reinitiation site because there are some after the Y122X initiation sites potentially. So
That's
I
right.
guess
Yeah.
it's not the same. It's not the only mutation. Did you try other mutation as well?
That was the only kind of early nonsense mutation that we tested, Y122X, and we only evaluated that in the 16 HBE cells. Yeah, we haven't tried anything even earlier, which there are several more of interest there. But yeah, just the Y122X, and yeah, that's right. That's what we observed, which was interesting because even though that nonsense codon seems to have a different branch regulation by NMD, it still can be the mRNA can still be upregulated when you target with SMOG6 ASOs. So that's why we propose the branch specific SMOG6 inhibitors might be a good pan approach for all CFTR mRNA nonsense codons.
So other questions from the chat. How are the ASOs delivered, and how can they deliver to submerged or highly differentiated cells? Because
Yeah. So the way that these experiments were performed is the ASOs were just delivered into the cell culture medium, and the ASOs were taken up by the cells just by free uptake. And we did several time course treatments, and we found that 6 days with ASO treatment was sufficient for knockdown as well as CFTR mRNA upregulation. Knockdown of the NMD factors was observed at much earlier time points, but 6 days was the good time point for the effects that we observed for CFTR mRNA upregulation following NMD inhibition. So the 16 HB cell treatments were at a submerged or a liquid-liquid interface. And then for the HB cells, they were air-liquid interface. And so we would deliver the ASOs from the basolateral compartment in the transwell inserts, and free uptake also for the primary HB cells.
okay, a last question, quick answer. How do your using chamber results compare to Y-type function in the cell lines? And I will ask, did you also try to add CFTR modulators to increase the function of
Yeah, so like a percent of wild type, perhaps. So the Y122X and W1282X 16 HB cells, there was a difference in the degree of upregulation of CFTR function following these treatment combinations, so the total wild type percent was probably between like 6 and 8% of wild type function. Pretty good, but not quite where we would want it to be. And so yeah, perhaps combining with the CFTR modulators specifically for the 1282X mutation, we would see even a further enhancement. And we have done those in-house, and we do see like a pretty good further improvement there. But that wasn't included here in the presentation.
So thank you very much, Eddie, for all these answers in this wonderful presentation. And now we move to Batsheva Kerem. So I will have to introduce Batsheva Kerem, but I don't know if I have to introduce her because everyone knows Batsheva Kerem. She's one of the discoverers of the CFTR gene, and she will speak about very, very interesting results. Antisense oligonucleotide splicing modulation as a novel CF therapeutic approach for the W1282X mutation. Thank you, Batsheva.
Hello, everyone. I am Batsheva Kerem from the Hebrew University in Jerusalem, Israel. And the title of my talk is Antisense oligonucleotide splicing modulation as a novel CF therapeutic approach for patients carrying the W1282X mutation. Here are my disclosures. I'm the CSO and the scientific founder of Splicing, and I will speak today on behalf of Splicing. As we all know, CFTR modulators are not yet serving all CF patients, and there is no available therapy for patients carrying various mutations, including nonsense mutation. Thus, drug development is essential to address the unmet need of non-responsive mutations, which includes also the W1252X nonsense mutation. The mutation resides in exon 23 and is classified as a minimal function mutation associated with a severe disease form. As a nonsense mutation, it leads to degradation of the transcript carrying the mutation by the nonsense-mediated messenger RNA decay. As a result, there is low levels or no levels of CFTR protein in Western blots, and accordingly, no CFTR function. So our suggestion is a novel antisense oligonucleotide-based therapy for patients carrying the W1252X mutation by skipping over exon 23. And in order to understand the molecular basis of this approach, let's look at the CFTR. And as we all know, there are 5 different domains in the CFTR protein. NBD2, which is the last domain, contains exon 23 with the W1252X mutation. CFTR biogenesis starts with co-translational folding of nascent polypeptides in the ER membrane. However, NBD2 is folded post-translationally and is not required for stable assembly of the protein and thus escapes the ER quality control. Several years ago, CFTR proteins lacking the entire NBD2 were shown to be stable and generate functional proteins. So our question was, can CFTR proteins lacking only part of NBD2, in this case exon 23, can they form functional CFTR channels? And this may be possible since exon 23 is in frame and therefore can be removed without causing frame shift of the open reading frame. Skipping over exon 23 bypasses the NMD of transcript carrying the mutation. And this is another advantage of this approach. So as a proof of concept, we have generated a CFTR cDNA lacking exon 23 and analyzed the function of proteins generated from this cDNA in the Flipper assay in HEC293 cells. Here is an example of such an experiment, and here are the quantitation, and we can see that CFTR proteins lacking exon 23 have low activity level that can be augmented by CFTR modulators, reaching in this system about 50% of wild type. So our approach is based on antisense oligonucleotides, which are RNA-like short sequences that can bind and modulate the RNA. They are specific to the target mutation region in the RNA and thus avoid off-target effects. They are chemically modified for stability and increased cell uptake and were clinically validated for several genetic diseases already. Using an in-house algorithm, we designed and synthesized antisense oligonucleotides in order to skip over exon 23 and tested their effect in 16 HBE cells genetically engineered by the CFFT to carry the W1252X mutation. We tested close to 80 consecutive antisense oligonucleotides and analyzed their effect first on skipping of exon 23 by RT-PCR. Here is an example of such an experiment. The control transfected ASO had no effect, all transcript carried exon 23, whereas several antisense oligonucleotides, like these ones, were highly efficient and promoted a very high level of exon 23 skipping, whereas others had a very low effect. So out of these ASOs, 39 were further analyzed by real-time qPCR, and 16 of them, here in green, were positive as they reached over 50% exon skipping. Some of them were very efficient. We then analyzed the effect of these 16 ASOs on the production of mature CFTR proteins. Of course, they are exon 23 deleted. So we can see here a Western blot showing the effect of some of our antisense oligonucleotide. Control ASO did not generate any CFTR proteins. Antisense 2, 3, and 47, for example, as can be seen here, generated a significant level of mature CFTR proteins. Quantitation of the effect of all of these show that some ASOs reach a very high level of over close to 50% of wild type. Our next challenge was to identify ASOs for patient heterozygous for the W1282X mutation. And by this, I mean that we search for antisense oligonucleotides that have the best effect on the W1282X allele, but the least effect on non-W1282X alleles. So here is an example of the experiment that we performed to identify such ASOs. We treated cells, 16 HBE cells, that carry either the W mutation, Y-type alleles, or F508 del mutation. In pink, we see the result of treating these 3 cells with different ASOs. All of them had a very high and efficient effect on the W1282X allele. You can see here they reached almost complete skipping over exon 23. ASO 47 was also efficient in skipping over exon 23 in wild type and A508 del cells. However, ASO 2 and 3 had a much lower effect, which was below 30%, and thus these ASOs were considered as specific for W1282X mutation. Next, we analyzed the specificity on the CFTR protein level. And we can see in this Western blot that the same ASOs that show a low level of ASO skipping in wild type and F508 del cells show in wild type cells specificity at the protein level. We did the same in F508 cells and got the same result. So we see with ASO 2 and 3, no production of deleted exon 23 proteins. However, with ASO 47, we see generation of B-tag protein. So this is a non-specific ASO. The specific ASO were further analyzed for their effect on the CFTR function. The analysis was performed in Nasal Chapter. We can see an example of such traces. Control ASO plus Trikafta generated low CFTR activity, whereas treatment with ASO2, for example, plus Trikafta generated a significant increase in CFTR activity. And the CFTR activation was dose-dependent, both for ASO2 and 3. And interestingly, ASO2 reached a median activation of about 20% of wild type which is very significant. Splicings and products are planned to be inhaled, given to patients once a week, thus reducing patients' treatment burden. For a successful treatment with inhaled aerosols, they should be stable and properly be distributed in the lungs of CF patients, which as we all know, are filled with mucus. So in order to investigate these requirements, we performed a battery of many experiments in vitro and in vivo. I will show just one in vivo experiment in which wild-type and beta-inox mice were treated intratracheal with our aerosols. Beta-inox mice served as a model for CF-like mucus. We stained the aerosol with a labeled sense aerosol and followed the distribution. So as can be seen here, the aerosol was distributed in wild-type and in beta-inox lungs very efficiently. We can see a wide distribution and is similar between the two types of mice. Taking a closer look into cross-section in different areas of the lung, we can clearly see distribution and, importantly, penetration of the aerosol into epithelial cells in wild-type and beta-inox mice in the different regions of the lung. So this is very encouraging. In the last slides, I want to present new results, which are quite preliminary, not yet optimized, analyzing the effect of our aerosols in primary human nasal epithelial cells from patients carrying the W1252X mutation. Aerosol was delivered by free uptake. So first we analyzed the effect on the skipping. And as can be seen here, both aerosol 2 and 3 promoted skipping over exon 23, which was dose dependent. Then we analyzed the effect of these aerosols on the CFTR function in the H&E cells. And as can be seen here, for example, treatment with ASO2 and Trikafta led to a clear activation of the CFTR channel, which reached twofold increase of CFTR function relative to control ASO. A very similar result was achieved with ASO3. We extracted RNA from the same cells that were analyzed in the Using chamber measurements and found that for both ASO2 and 3, 50% of the RNA missed exon 23. So skipping was about 50%, which is quite high. So altogether, our results show the high potency of these lead ASOs for patients carrying the W1252X mutation. And it is important to emphasize that currently these patients have no available therapy. So in summary, our lead ASOs promote high levels of exon 23 skipping, generating mature CFTR proteins in cells carrying the W1252X mutation. These ASO significantly restore CFTR function. They are well distributed in lung and penetrate into epithelial cells, even through mucus. And Splicing aims to initiate clinical studies in 2022. So I would like to thank the people who did all this work, the Splicing team. Here we are in Jerusalem Hills. I would like to thank our close collaborator, Eitan Kerem, from Hadassah Medical Center in Jerusalem, and Christine Baer, and her former postdoc, Onofrio La Selva, from the Hospital for Sick Children in Toronto, Canada. And thank you for your attention.
Thank you, Batsheva, for this wonderful talk. So we have many questions now on the chat. The first question is, assuming that you need some kind of modulators, and you show that on the 16-HBE and the nasal cells as well, would it be combo or only potentiator?
Yeah, so the potentiator we tried, VX770, we did see an effect, but it was a small effect. So at the moment, we think that probably Trikafta will be needed to achieve a significant effect for the patients. Yeah.
Okay. And second question, very important. So did you confirm the specificity of your assays in other mutations? I should say so, wild type, homozygous, and I should also ask other nonsense mutation in a wide range of mutations.
So yes, we tried specificity in H&E cells and found that it is really, really specific, even if we go very high in the concentrations. And as we are using antisense oligonucleotides specific for the W, we did not look at other mutation rather than F508 alleles or W alleles. But we got the same result. And if exon 23 is the wild type sequence, we don't expect any difference.
And so how many mutations can we find in exon 23? Because that might be also interesting for the other mutations.
Yeah, there are several mutations in exon 23. The number of patients carrying each of the mutation is very small. Yeah, so if we will design antisense oligonucleotides specific for these mutations, it can be of help for the patients. Yeah.
So did you also assess which cells do you target? So in your primary cultures, because the primary cultures as well as in vivo are the nasal, the bronchial epithelium is composed of ciliated cells, but goblet cells, etc. Did you assess which cells your assay are going to penetrate in already in your mouse model, for example?
Yeah, in the mouse model we did, and we can see actually uniform penetration in the epithelial cells in all of them. We didn't see, but it's not a single cell resolution, okay? But we do see them in all type of cells.
So a question, how do you think the specificity is being conferred? So is the on the molecular basis, I should say, is the assay overlapping with the W82 sequence?
Yeah, they are. Yeah, both lead antisense oligonucleotide number 2 and number 3 are on the mutation, and it's amazing to see that only one base pair change from a wild type sequence matters, as we know, for PCR primers, etc. Yeah.
Do you have a correlation between the level of exon skipping and the rescue of the CFTR activity?
Yes, it is dose dependent. Yeah, as we go higher, we see, at least on the RNA level, we see more skipping. And we are now doing these experiments in H&Es from patients, right? What I presented is only the first patient, which was just not optimized yet, and we are working on expanding it. Yeah.
So, and the last slide, so the very interesting and encouraging preliminary reports on the nasal cells show some rescue, but so as far as I guess, so it's 20% of wild types. So how much wild type CFTR activity do you rescue on the nasal cells?
Yeah, so it's early days. I would not say a percentage because it was one patient, and the antisense oligonucleotide concentration was not yet optimized. It was significant, but it's early days. Let's wait.
Okay, and so do you assume, because now thinking about translation to humans. So, so what do you think it will be well tolerated? Because you show some, some interesting data for the future. So do you think it will be so once a week, as you suggest? And so on the long term, will it be well tolerated? What would be, this tolerance?
Yeah, so splicing is studying this now in mice, and we are soon will start experiments in non-human primates. So in order to answer your questions, and yes, we hope that we will need only once a week administration by inhalation. This is since ASOs are very, very stable. The only question is whether cells are proliferating, dividing, and then you get less ASO after the division in each daughter cell. That's the only concern. The antisense itself is very, very stable. So, yeah.
And so do you anticipate that if the patient is compound heterozygous, so having another mutation on the other allele, do you think that you might give less ASOs as it is concentration dose dependent? If you have only one allele to rescue, I should say?
Yeah, we will have to look into this. And this is exactly what we are doing now with the H&Es, right? So the future will tell, it's too early.
Okay, and so I have another question as well, waiting for questions from the assistants. What is the stability of the SO in the environment of the CF lung? Inflammation, infection, etc. Did you study that already?
Yeah, so the only experiment for now that we performed was in INAC mice, right? They have a CF-like mucosa and some of the features that we see in CF, and the ASO penetrated very, very efficiently. But we will have to wait for the clinical trial, as we don't have animal models with the mutations, so we'll have to wait. And we are quite optimistic. We'll see.
So we wait for you next year, I think, with new results. So thank you very much, Batsheva.
Thank you very much.
Thank you. Now we move to another topic, very interesting, about gene therapy, which will be presented by Dr. Farrow. Dr. Farrow is postdoc now in Burlington University, and he was PhD in Adelaide, and he worked on airway stem cells, made his PhD on airway stem cells, and he will now speak about neonatal airway gene therapy delivery to enable effective adult dosing. Based on a lentiviral virus vector. Thank you very much.
Hello, my name is Dr. Nigel Farrow. I'm currently at the LUNAR College of Medicine at the University of Vermont. I'd like to thank the Cystic Fibrosis Foundation for the opportunity to give this presentation. This is some work I did recently with the Adelaide Cystic Fibrosis Gene Therapy Group based at the Women's and Children's Hospital and the University of Adelaide in South Australia. Today's talk will be titled Neonatal Airway Gene Therapy Delivery to Enable Effective Adult Dosing, a lentiviral vector study. On some housekeeping, I have no relationships to disclose related to this presentation. Okay, some basics first. We know that cystic fibrosis is a lethal monogenic disorder which affects approximately 1 in every 2,500 newborns. It is caused by mutation or mutations in the cystic fibrosis transmembrane conductive regulator gene, leading to a loss of chloride channel in epithelial cells. This then goes on to disrupt the trans-epithelial ion transport and subsequently the function of organs which align with epithelia. This leads to malabsorption of the digestive system and brachioestasis of the respiratory tract. And while there are currently treatments that can alleviate the digestive symptoms, there is really no effective treatment for the respiratory tract. This leads to a poor quality of life and an early mortality for those who are unfortunate to have this disease. In research done in Australia at the Cystic Fibrosis Airway Research Group. We focused for about 2 decades now on gene therapy approach, focused at the airways. And that is, the basic premise there is to insert a functioning copy of the CFTR gene into the airway cells. And this has the potential for correcting the underlying cause of CF airway disease, really at a molecular level. A lifelong correction is going to be required and a one-off treatment is desired. So to achieve this, our gene therapy program has really been centered around a lentiviral vector due to its ability to instill its genes into the genome of the recipient. We use a HIV-1 based lentiviral vector with a BSVG pseudotype. Over the course of the last couple of decades, we've developed a 2-step protocol in which we form our gene therapy. And that is to first instill a dose of lysophosphatidyl or LPC, which essentially opens the tight junctions between the cells, allowing for the vector to gain access to the receptors that lie on the basal electric surface cells. 1 hour after this preconditioning treatment, we then instill a dose of our lentiviral vector. And just to give you some background on what we've managed to achieve so far and where we're headed, we have shown that from a single dose of our gene therapy vector, we can show through marker genes that they are being expressed for up to 92 days in a mouse, and more importantly, proof of principle with a therapeutic gene for at least 110 days, showing at least partial correction. We then expanded that study out and again repeated a very similar study, but took the time course out for even longer. And we there showed that a single lentiviral vector dose could produce expression of the selected marker gene for 24 months in mice, which is essentially the lifetime of the animal, which was very exciting. And again, more to the point is we show a therapeutic gene was delivered and was expressed, showing partial expression for at least 12 months. Well, we've published a number of further studies over this period. Recently, we moved to non-human primates and have successfully shown that we can again get the marker gene. Both luciferase and LZ were expressed for a month, which was the time of that study. And most importantly, we showed that we could indeed get the human CFTR gene to successfully be delivered through our vector, integrated into the genome of a normal non-human primate. However, moving forward, achieving consistent high and sustained levels of transgene expression is essential but remains challenging. And we feel this is not only for our group and our vector studies, but the other groups that have also been doing lentiviral vector work around the world. So we set about this pilot study and to try and improve on our previous results. And that was to determine if delivery of a lentiviral vector during the neonatal development facilitates reliable and or higher levels of transgene expression from subsequent adult lentiviral vector dosing. So the basic premise here is we wanted to see that soon after birth, we're using rats in the study, soon after birth, when the immune system is still essentially setting itself, if we could basically fool the immune system or the animal systems into believing that the transgene was self and would not have an immune response down the track, and basically give us a more reliable outcome. So to undertake this pilot study, what we did, we took 2 groups of Sprague-Dawley rats, a control group and a treatment group. And at 3 days of age, when they were about 5 or 6 grams by memory, we treated the treatment group with 10 microliters of the BSVG lentiviral vector. They did not receive the LPC pre-treatment. And the vector contained a luciferase transgene, which was driven by the EF1 alpha promoter. The control group received a PBS sham at the same time. And then 6 weeks after the initial nasal dosing, we performed bioluminescence imaging on all the rats. And we also took serum samples obtained from all animals at the time of BLI. So from that first imaging time point, you can clearly see here that in all animals, there was strong luciferase expression. And in some of the animals, it was indeed very, very strong indeed. We've got to remember that these rats now are about 150 grams, and they were dosed and they're about 5 or 6 grams. And it was quite surprising and striking to see just how large of an area was being expressed, indicating some further interesting things that to be explored in other studies. So after this bioluminescence imaging assessment, the now adult rats were treated with an adult dose of our gene therapy. And to do that, we used 25 microliters of LPC for our pre-treatment, followed 1 hour later with 50 microliters of our lentiviral vector. And they was delivered by a miniature bronchoscope into the right main bronchus. So essentially we went down through the larynx into the trachea until we hit the carina, and then guided it off into the right main bronchus, where we delivered both our pre-treatment, followed later by our vector. We then, 1 week after this adult treatment, performed bioluminescent imaging again, just to examine the levels of expression that were apparent both still in the nasal airways and in the lungs. And again, we took serum samples from all the animals at this time while they were anesthetized. So coming to the nasal first, you can see that there was indeed still a good signal from a lot of animals, the luciferase gene being expressed. It is appearing to be somewhat waned. However, when we compared this through statistical analysis to the first imaging time point, there was no statistical difference in the levels of gene expression on average. And what we're seeing here, we believe is more that in the two weeks between the imaging time points, these animals had grown some up to 50 grams. So they were at that period in life where they were really putting on weight. And we believe that that was blocking part of the signal coming through. Most importantly to this study, if we have a look at the adult dose into the lungs, if we look at the top panel of animals here, listed from C1 through to C10, the control group, there was 6 animals clearly had luciferase expression from very bright in control animal 9 to quite low in control animal 8. And indeed, 3 of these animals had nothing in the lungs, or 4 had nothing in the lungs, with 1 of those having it in the trachea. But if we move down to the treatment group, you can see that there is a more reliable pattern here, and 9 out of these 11 animals all had quite decent luciferase expression, with treatment animal number 11 here showing quite outstanding response, and really coming out of that right main bronchus it was focused on. So we wanted to compare this and see exactly from the animals that did express luciferase whether the actual levels on average were improved in the treatment group. So we assessed this through a fitted linear mixed model to the log BLI, and we showed that despite the indications of improvement in the treatment group and a more reliable outcome, there was no statistical difference in the mean BLI between the control and treatment group after an adult LV lung dose. However, I will point out that the p-value here was 0.055, so it was very encouraging, and we believe this needs to be followed up. So the serum samples that were taken were assessed for an antibody response, primarily to the luciferase transgene so far for this study. And they were compared from the control and the treatment group to another group of animals which had essentially received no pre-treatment, no LV vector whatsoever. And we found that there was no antibody response present to the transgene as observed in either the treatment group or the control group. So in conclusion, our results suggest that this form of neonatal dosing does not increase overall acute lung transgene expression levels when compared to a control group. However, again, the p-value of 0.055 suggests that as a pilot study, this is not definitive. Also, the number of non-responders in the neonatal treatment group compared to the control group suggests that the delivery of the LV vector at a neonatal age will result in a more reliable outcome of successful transgene expressing animals. But again, future studies with a larger sample size are really required to tease this out. And also in this future study, we plan on doing a more comprehensive and in-depth assessment of the immune antibody responses against both the transgene and the lentiviral vector, just so we can try and get more of an understanding of exactly what's at play here. I'd just like to acknowledge the Channel 7 Research Foundation of South Australia, who primarily funded the study, and the MS McLeod Foundation and Cystic Fibrosis SA, who supported myself as a research and other members of the team. Also, the University of South Australia and South Australian Women's and Children's Hospital for supporting us in all of our research. Thank you very much.
Thank you, Nigel. This was very, very interesting and very interesting results as well. So I will begin with the first question. So your results indeed suggest that this form of neonatal dosing does not increase overall acute lung transgene expression levels. Do you have an explanation for that?
At this stage, no. It's taking from the individual animals, it really didn't raise things too much as we had hoped. And I think, I feel a larger study with more animals, we may have seen a difference. It may go the other way as well, but we are very excited to push forward and repeat this study with a lot larger group of animals, and indeed do it in our newly formed CF rat.
So, apart from increasing the number of animals, any change in the experimental conditions, the timing of administration, etc.?
Great care was taken through this to ensure everything was done exactly for each animal, and we've had a lot of experience in doing this. So when we look at not just within this study, but from the group that got the neonatal dosing, when we look at previous studies we've done over a long period of time, there was clearly a more reliable outcome. So I feel over a lot of independent studies that were looking at gene therapy dosing to the lungs and indeed the nasal airways, we've always had that from very good result to animals with nothing and then everything in between. But we did find the actual number of responders, or I should say, compared to the number of non-responders in this, there was clearly a different scene.
Okay, thank you very much. So a question from Maria Limberis. So may I ask if your experiments suggest that there is a difference in permeability between the adult and the young or the newborn epithelium?
Well, with that, the newborn rats that were dosed, they were due to animal ethics and the sort, we didn't anesthetize animals for safety reasons. They were dosed while awake. Therefore, the decision was made to not give the LPC pre-treatment to open the tight junctions, which is where I can see this is going. It's a tough one, that, because we did see, as you saw, a great response from those animals that were treated without the LPC, which normally if we don't use LPC, we don't really see that in adult mice. All I can suggest is a stem cell of some sort was hit and has been passed on, which is why there's such a large expression. If that was indeed because it got through a tight junction in the younger animals or not, I really don't know. That is a great question, Maria, and it's something I think we need to look at is why indeed we got through that tight junction barrier without the use of our pre-treatment.
And as well, did you also investigate the effect of the inflammation on your gene therapy? Because we know that inflammation begins very early, including infection as well. We know that from the ferret model, it's very important. So did you consider that as well?
It's definitely part of the consideration, and seeing the response we got from the neonatal period, it's opened up the pathway for other studies, which we really would like to look at the effect of overall dosing in the lungs and the sort in neonatal animals prior to the inflammation that is caused in the lungs. And as I mentioned before, the group in Australia have a very good CF rat model. And so there's other experiments going on looking at the onset of disease and inflammation in those animals. And when we've pinned that down, we can look at seeing if we can dose prior to that and prevent this. So I think it's there's some exciting things ahead.
So yeah, this rat model, this Australian rat model, does it has a CF-like disease? Because as far as I remember from the publication, there was not.
You are correct. However, we're looking at, as has been achieved with some of the other models, whether or not that can be induced through infection models into these lungs. And if we can achieve that, then we can go back a few steps and look at preventing it in those situations.
A question about the immune response, because you say there was no evidence of immune response, so should say a systemic response. So did you have any evidence of, so you show there was no difference in weight, but did you have any evidence of other organs change after your treatment, after organs targeted?
Nothing that was observed. However, as a pilot study, we really wanted to just have a look at what we could with these serum samples. These serum samples are still being investigated as we speak, leading towards the publication. Again, I'd like to repeat the study with a larger group of animals and indeed include a lot larger section in that study to look at overall immune response, both systemically and throughout the body.
Do you think, it's a very naive question from me, because you work in the rat, do you think you may have a species specificity working on other animals, or no possibility of that?
Sorry, I didn't quite understand the question.
Do you think you might have some different results if you work in the ferret or in the pig, so species effects?
That's an interesting question. In the past, we have done studies in normal ferrets, which we were doing in Australia leading up to CF ferret models, but it's incredibly hard to bring these animals into Australia. We decided to create our own CF rat, which is why we went that direction. Our published studies in the ferret were very promising. In the past, we've also done sheep, which were very promising. Marmosets as non-human primate, again, very promising. And I do believe currently in Australia, the group are looking more at pigs and that sort of thing. So at this stage, we have found generally across the board, whichever animal we go into, whether it's looking at proof of principle in the small rodent models through to the larger airways of a sheep or a more human-like airway of a non-human primate, we have found encouraging results across the board.
Thank you very much. Thank you very much, Nigel. This was very interesting for all the answers for the questions. And now we will move to our next speaker.
Thank you.
Christina Barillia. Christina is postdoc at UCL. She has worked on DNA editing for CFTR, but now she will present also very novel results on the development of iPS-based airway epithelial platform for evaluating patient-specific responses to modulators. Thank you very much, Christina.
First of all, I want to thank you, the organizer, for selecting our poster and give me the opportunity to share our data with you today and all of you that are interested in this talk. My name is Christina Barilla, and today I'm going to show you some of our effort in the field of the personalized medicine. I don't have anything to disclose for this presentation. First of all, I want to remind you that we already have more than 300 causative mutations that are associated with the cystic fibrosis, all in the CFTR gene. For the purpose of this presentation, I categorize them in 3 groups. In the first one, we have mutations that do not affect the synthesis of CFTR, but its function or maturation. For this mutation, we already have modulators that are approved for the treatment. In the second group, we have a similar type of mutation, but because they are either rare or not well characterized yet, we don't have approval for the treatment yet. In the third group, we have either splicing variant or premature termination codon mutation. which we do not expect to respond to modulators, either because they cause no production of CFTR or a really few amount of functional CFTR. Actually, currently the field is mostly focused on the third and the second group in order to extend the treatment to all CF patients. But we need to remember that even in the first group, we have a patient that do not respond as we expect to the modulators because of their genomic background. They may have, for example, a non-causative mutation in the CFTR or in other gene that act as a modifier on how the modulators affect on the CFTR. So to extend the treatment to all CF patients, we of course already have some model that we can use, but for our specific interest and the purpose, we are really interested in the personalized side of this treatment. And in order to do this, we really want to have patient-derived samples. So for our purpose, the immortalized cell line may not be suited. Also, our main interest is how the modulators would benefit the patient at the lung and the airways. So this would potentially limit our application for intestinal organoid, and we may be more interested in primary cells. But this would also cause us some limitation in terms of how many patients we can actually study or how many cells we can get to perform the study that we need to do. So for our purpose and in our vision, we want to have what is the tissue that we are interested in a study, and in this case it would be the airways, and also deriving from a patient with its specific genetic background. So that's the reason why we are currently mostly interested in the use of iPS-derived airway cells. We now can use this model because we established a reproducible and a reliable protocol to generate these iPS-derived airway cells. In the last several years, we collaborated with the Boston University, in particular with the group of Drs. Hawkins and Cotton, to develop this stepwise differentiation protocol, which consists in 3 main steps. The first is the induction of a lung progenitor, which we now know that we can enrich through surface markers such as CPM. We then induce the airway progenitors, which then maturate into iPS-derived basal cells, or IBCs as we call. These cells can be selected and enriched through NGFR and EpiCam positive. We already showed in this recent publication that these cells are able to give rise to pseudostratified epithelium in ALI culture and generate a readable and consistent CFTR current that we can analyze at the Assay chamber. Despite this protocol is really consistent, robust, and reproducible, we noticed that by extending the number of lines that we differentiate, we sometimes have some of the lines, such as the 2 in the center that I'm pointing at now, that do not perform as efficiently in the induction of IBCs. So we sought to find some surface marker that could enrich the airway progenitors around the day 30 time point. In particular, we selected F3 and the EGFR, which we saw it highly enriched NKX 2.1 population in this time point. And by introducing this additional sorting step, we further improved our capacity to induce IBCs, in particular for those iPSs that wouldn't be as efficient as most of others. More importantly, if we introduce this sorting step And as you can see, in particular, in cells that perform less efficiently in the induction, they still maintain their capacity to differentiate into pseudostratified epithelium in ALI, and they can be read at Acin chamber analysis. What we want to do with these cells is to use them as a tool to predict what would be a patient-specific response to a given treatment. To do this, we first applied the protocol to IPS line with the genotype we already know to be either responsive or not responsive to a specific modulator or treatment. The first example I'm going to show is a heterozygote IPS line which carry 349del-TT, which cause the formation of a premature termination codon, and we do not expect to respond to the modulators. On the other allele, we have the G551D, which we know, and it's already approved to the treatment for VX770, and more currently for the triple combination therapy. On the left side, I'm showing the Acin tracing, where each trace with different color represent the same IPS line, but treated with a different compound for 48 hours prior to the Acin analysis. With arrow, I'm showing a specific time point of acute treatment during the tracing. On the right, I'm showing a summary of the data from the same line, which was obtained from 3 independent biological replicae, and each of them had a technical replica for the Acin analysis. As you can see, both the chronic and acute treatment show an increase of the CFTR current, in particular the one derived from the forced choline induction, and these even further improve when we treat with the triple combination therapy. This further improvement was already observed by Dr. Raab in the FRT model, and he showed that last year in NACFC. The second example is a heterozygote line which has a splicing mutation and a stop codon mutation, both of which we expect to have little or no functional CFTR, which we can see also from the summary of the Acin data, and they do not respond to the triple combination therapy. But if we treat with the NMD inhibitor and the read-through molecule, which should rescue at least the production from the G542X, we see that we have a modest but consistent increase of the CFTR current. I hope I convinced you that we generated an even more robust protocol to obtain IBCs and highly pure IBCs, which are responsive during electrophysiology analysis, at least during the Acin chamber, with the high consistency. I didn't show details on the clone-to-clone on the experiment-to-experiment consistency, but if you are interested in, you can find the example at our poster. Most importantly, we are observing a response to specific modulators that are consistent with what we would expect from a known patient genotype. So now what we can do with this protocol and the cells is, for example, study modulators in the context of rare mutation or mutation that are not approved yet for their use. Like, for example, in the case of N1303K. We know, and other group already reported, that this mutation responded to VX61 and the VX770, and also we can observe in our line But if we treat with the TCT, this improvement is even greater, even though the current itself is modest. We also are aware of other groups, which you can also see at the workshops or poster in this conference, such as the poster of Dr. Brewington, that is the number 638, which is obtaining a similar response to the triple combination therapy in different models, supporting the idea that we could extend the treatment to this mutation. Of course, further study need to be performed, but this would support this conclusion. Another possible application is related to the clinical trial. We could, for example, study CFTR mutations that are extremely rare, which wouldn't allow to have enough patients to create the clinical trial with a significant number for both control and treatment group for good statistical significance. We also could study patients that have a CF phenotype but don't have a molecular diagnosis yet. We could use these protocols also to stratify the patients that have the same mutation, but at least at the IPS level, they have a different response to the modulator, so that we could increase the statistical significance of the response at the clinical trial. The latest example I have is, for example, to study completely new modulator or treatment for in the future. Before being able to do this, we will wait the result from our ongoing clinical trial, which aims to correlate the response that we see in vitro in iPS cells to the actual benefit that the patient have. Some of the preliminary data are already available at Dr. Linnemann poster 572. Lastly, I want to thank our collaborator. This is a combined effort between us at Houston, the Emory University, and the UAB. And also I want to thank all of you for your attention and your interest in this work.
Thank you very much, Christina. Very, very interesting results. So we have some questions. The first one is, what do you think is the advantage of going through this lengthy and I should say costly process of also of generating and differentiating iPS instead of using nasal cells or intestinal organoids? Which are the advantage of your model?
This compared with the nasal cells, which are primary cells, we definitely have the advantage that we can expand a lot more, so we can have a lot more testing possibility in terms of type of modulators or a number of the experiments that we can replicate. Also, with the nasal cells, we may observe some variability on how they are cultured between different labs, and we may have more limitation in the sampling, while the iPS we can generate from anything. It can be like a skin biopsy or from the blood too, so it's not necessarily too invasive. Regarding the intestinal organoid, as I mentioned in one of the slides, our concern is that the level of the CFTR expression may not be exactly representative of what we see in the airways. So we are worried that we may overestimate the benefit that we would observe in the patient.
And so, how many your cultures, your airway cultures, how many passages do you use? So, or can you really have a lot of messages, 10, 20, doesn't vary for CFTR and other transporter expression? Did you study that?
For this specific project, we directly sort for NGFR and EpiCam and plate for the analysis. But in the original paper on the BO3 line, we expanded as an organoid for over 3 or 4 months. And the IPS itself, before the differentiation, can be expanded also for a long time, like passage 30 or even more before the differentiation. And we didn't test the long expanded IPSS in this protocol, but we tested the CFTR expression on the long expanded organoid, and we didn't see a dramatic effect on our ability to read the CFTR current in the assay.
Thank you. A question, did you analyze the effect of the modulators in cell scarring and N1303T W1282X?
Yes, we had one line with that genotype, and we observed increment when we have the triple combination therapy. So far, we only tested that specific patient, but we are planning to test more with the NSL303K mutation.
And so, did you study different clones from the same patient? Because it's in your conclusion. So, but that's really very, very interesting. Also to understand which factors regulate the different responses in patients having the same genotype. So this would be a very, very interesting model to study this.
Yes, in our hand, we had several lines that we tested 2 clones, and we see consistent result in those. We don't see much variability based on the clone so far.
Okay, so same level of mRNA, same level of CFTR activity, same level of correction.
We didn't try the correction, but on the assay chamber, yes, we see the similar current between the 2 clones. And also, if we do biological replicate within the same clone, that we still see consistency in between the 3. So it seems to be really affected from the genotype, not the preparation or the IPS clone itself.
And did you resample the same patient? If you resample the same patient, you obtain the same, not the same clone, I should say, but the same type of culture?
We didn't try that yet, but we have in a program to test both skin biopsy and blood-derived sample from the same patient. So hopefully we will have an answer on that soon.
Okay, thank you very much. So your future development plan are to have more results, I should say, more mutation. And so to correlate to that, to the in vivo response, so in patients treated with Trikafta, to link that to the level of the response. So what are your next plans?
Yes, right now we have a collaboration with the Emory University and the UAB, and they are in parallel testing some of the patients that we are also differentiating in IPSS to see if the response that we see in vitro is corresponding to some improvement on the CF phenotype. So far, we only have 1 patient that we have the data from both, which is encouraging, but we will definitely need to have information from more. And this is ongoing. As I mentioned, you can find some preliminary data from the Dr. Lindemann poster.
Thank you very much. Thank you very much, Christina, for all these answers. So, and we move to our last presentation now from Dr. Perez Garcia. So he is part of the LUNAR CF program, and he will speak about mRNA therapeutics to correct the root cause of cystic fibrosis lung disease. Thank you very much.
to the organizers for the invitation to present today in this workshop. The title of my presentation is LUNAR CF, a novel mRNA therapeutic approach to correct the root cause of cystic fibrosis lung disease. All the data presented today will also be available in poster 549. This is the forward-looking statement, and this is my disclosure. I'm an employee at Arcturus Therapeutics. LUNAR CF is an mRNA replacement therapy to treat the underlying cause of cystic fibrosis lung disease. We custom optimize the human CFTR mRNA that will produce a human CFTR protein, and this mRNA will be encapsulated in LUNAR, our lipid nanoparticle delivery system. With the use of a nebulizer, we will aerosolize the LUNAR formulations directly to the airways of the CF patient. This is a mutation-agnostic approach. The encapsulation efficiency is maintained, as we can see here in the plot, where we measure the percentage of mRNA that is still encapsulated in the LUNAR nanoparticles after nebulization. We use different concentrations of LUNAR and 5 different types of nebulizer color-coded here on the plot. Overall, the encapsulation efficiency was maintained across all the nebulizers tested. LUNAR also shields and protects the mRNA in CF sputum. We generated 9 different LUNAR formulations carrying an mRNA, and we also generated the naked mRNA version. When we incubate them in a sputum derived from 2 CF donors, and we quantify the levels of mRNA after a period of incubation, we observed that the mRNA was preserved only in those conditions in which the mRNA was encapsulated in LUNAR. On the contrary, the naked mRNA was fully degraded. The droplet size of aerosolized LUNAR particles are within the respirable range. The activity of the mRNA is preserved in the aerosolized formulation, as we can observe here with a LUNAR formulation carrying an EGFP mRNA. When we generate an aerosol using a nebulizer and we compare the levels of EGFP expression in both post and pre-nebulization fractions, we can see that the levels are comparable. When we aerosolize LUNAR, in this case using a luciferase mRNA to the airways of a rat, and we measure the luminescence generated by the luciferase, we can see a highly deposition in the airways as expected. The delivery of LUNAR formulations to airway epithelium has been achieved across multiple species, and the slide is a summary of that. On the top panel, you see 2 rodent models, the mouse on the left and the rat on the right. And in the bottom panel, you see 2 examples of non-rodent models, the ferret and the NHP. In all these animal models, we use a reporter mRNA encapsulated in LUNAR. When we deliver the LUNAR formulation to the airways of those models, and we use immunohistochemistry to unveil the expression of the protein, we do see a very nice distribution across the airways in all the animals tested. We have done several studies trying to characterize the type of epithelial cells that we are getting into with LUNAR. Here, I'm going to show 2 studies that were done in collaboration with John Engelhardt at the University of Iowa. In the left panel, we do see a study that was done using ferret bronchial epithelial cells derived from the ROSA 26 transgenic ferret. They were grown in an air-liquid interface, and they were dosed with LUNAR mRNA. The cells that uptake the LUNAR mRNA will strongly express EGFP, and this EGFP can be quantified. Here in the plot, we do see that around 40% of the total cells in the culture were EGFP positive. We can also determine the percentage of cells that are either ciliated or secretories but colocalized in the EGFP with specific markers for ciliated and secretory. As we can see here in the plot, around 25 to 30% of ciliated and secretories express EGFP. This data was confirmed by colocalization using immunofluorescence for EGFP, here in green, and specific cell type markers for ciliated and secretory. A similar study was done in human bronchial epithelial cells derived from non-CF donors. They were also grown in an air-liquid interface, and they were dosed apically with LUNAR TD tomato. As we can see here in the plot, around 32% of the total cells in the culture are TD tomato positive, indicating that the LUNAR formulation was transduced in those cells. When we colocalize with different cell markers for the different epithelial cell types, we can get an idea of the ratio of TD tomato cells expressed per cell type. For example, here in goblet cells, around 30% of the total goblet cells in the culture are TD tomato positive. This data was also confirmed by using immunofluorescence for TD tomato and specific cell markers for ciliated secretory cells. Functional restoration of the chloride activity was achieved both in vitro and in vivo. In this particular example, that was also done in John Engelhardt's lab at the University of Iowa, we used ferret bronchial epithelial cells that carry a G551G mutation. We grow the cells in an air-liquid interface culture, and we dose them with different doses of LUNAR CFTR. When we measure the CFTR-mediated chloride currents after induction with first culling, we do see that at the two highest doses tested, we do see a very nice dose response in terms of chloride activity that are comparable with the positive control ivacaftor. On the contrary, the LUNAR carrying a TD tomato mRNA at the highest dose did not show any activity, indicating that the activity observed is specific to the de novo produced CFTR. In collaboration with Martin Mann's lab and the CFF lab, we did a study using human bronchial epithelial cells that carry a homozygous mutation for the Delta 508. We grow those cells in an air-liquid interface study, and we topically dose them with different doses of LUNAR CFTR. When we measure the CFTR chloride activity generated after first culling induction, we do see a very nice dose-dependent increase of chloride activity that seems to be plateauing at the highest dose. The activity generated is significant. When we extract protein lysis from those wells and we run Western blot for CFTR, we do see a dose-dependent increase in the generation of the mature CFTR protein in the treatment groups that are not present in the LUNAR buffer, indicating that the CFTR that we are producing here is due to the de novo produced CFTR after treatment with LUNAR CFTR mRNA. When we determine the C to B band ratio, we can also see that we do see a 6 to 8-fold increase in C band respect to B band in this study. In collaboration with Craig Hodges, we ran some studies using the G542X CFTR mice, and we showed that we can actually restore the chloride activity in this model using the nasal potential difference. What we did is that we dosed topically those animals in the nasal epithelia using a LUNAR CFTR mRNA and also LUNAR buffer control. When we measure the nasal potential difference generated in treatment versus control, we do see a significant increase in the treatment group that was not observed in the control buffer group. Overall, the take-home message is that aerosolized LUNAR LNPs are stable, respirable, and shield the mRNA from degradation in CF sputum. Target engagement has been achieved to epithelial cells in both in vitro and in vivo studies. We have shown the optimized human CFTR mRNAs generate significant levels of mature CFTR protein. We have also shown proof of activity for LUNAR CFTR in vitro and in vivo. Overall, the data strongly support further development of LUNAR CFTR for the treatment of CF lung disease. I would like to acknowledge all the people that generate data for this presentation. On the Arthur side, to all my colleagues listed in here, and to all the collaborators, in particular, John and Xiaoming at the University of Iowa, Stephen Rove in the University of Alabama at Birmingham, Craig Hodges in Case Western, Martin Mans, Kevin Coate, Ji Zhen, and Jean Mahoney at the CFF Lab. And of course, thank you so much to the Cystic Fibrosis Foundation for the constant support in this program. With this, I conclude my presentation, and I will take any questions.
So thank you very much, Carlos, for this very interesting presentation. So we'll have a first question. Can you please tell us what was the level of functional correction in Delta 508 relative to wild type?
So in this particular studies, we didn't have a wild type control, but I can tell you that the levels of the doses tested were comparable to or can be. We need to run additional studies running additional modulators, in particular, including the Trikafta to see where we are, and also including a more relevant positive controls. But those were like preliminary studies that were run not a long time ago. So it's just an early piece of data.
You show that your compound is stable in the CF mucus, but as far as I understand, it's also in vitro experiments. Did you really assess that in vivo? For example, in a representative animal models such as the ferret with a lot of mucus production?
Yeah, we are actually running those studies in vitro and in vivo. In particularly, we're very interested in our collaboration with John Engelhardt on the CF ferret model, and we have planned several studies to trigger efficacy endpoints in which we will have the mucus layer pression in those animals. So we will be able to determine if our drug product can cross through the mucus and also have some impact on the functionality of the airways.
So do you think you need to combine that with other therapies of mucus fluidification, for example, to help this to penetrate in vivo?
We don't know yet. That would be the simple answer. Our LUNAR formulation has been designed to actually, with that point of view in perspective, to be able to go through the mucus. But we don't have any data yet that will let us to make that conclusion. Definitely, we will consider that option. That's for sure.
So a question about toxicity from Maria, and I will add something as well. So first, do you have any data about tolerance, inflammation, induction of your compound when you perform inhalation? And second, considering toxicity, do you think you are going to get supraphysiological level, I should say? So in other words, did you try that also in wild type animals to check if you increase the wild type CFTR function?
Yeah, so in terms of the toxicology, we haven't generated much data yet. We had a hard stop due to the COVID pandemic in terms of non-clinical safety tox studies. We just started to trigger them a couple of weeks ago, so we will have a better understanding on where we are in terms of tox of our formulations. For our ongoing clinical trials using LUNAR formulations for the metabolic disease, the OTC, and for a vaccine, the tolerability seems to be pretty much clean. We haven't seen anything that is a red flag, but of course, those are an IV injection, those are an IM injection. We still don't have an understanding of the level of tolerability due to an inhaled product in a lung. We don't know it yet. So hopefully in future presentations, we can clarify that a little bit more. What was the second question, Isabel?
The question is, do you get supraphysiological level, I should say? So did you check the LUNAR in wild type animals to check if you increase the CFTR activity above from the normal? And second, in CF animals, do you get activity above the wild type level?
So to the second questions, we don't know yet because we need to run those studies with a side-by-side comparison with wild types. So we're still working on that. I mean, we have been developing the delivery of lipid nanoparticles for air liquid interface. You know, it's something that it was very well established for small molecules, but not for lipid nanoparticles. And with the help of Martin, we have done a very systematic approach to get a better understanding. So we will be triggering some of those studies and some of those comparisons with other modulators and wild type animals. In terms of the level of activity, in terms of the data that we have generated, delivery wise in the different animal models, all this data has been done using reporters. And the reason is that it's a problem in the field. There's not really good antibodies for CFTR that can let us identify where we are and compare to wild type levels, right? However, we have been able to develop 2 assays that can discriminate human CFTR from NHP matrix and from a ferret matrix, and we hope to put in place those assays in the upcoming studies. So we'll get a better idea of where we are in terms of the protein levels, and also we will use a reporter cohort to help us also understand the delivery towards which type of cell types are we actually getting into.
Okay, last question. Short question, short answer. Could you review the short circuit current values on the plot next to the Western blot again? In other terms, do you have a correlation between CFTR activity and BANC rescue?
Yeah, one of the things that we are saying, and this is something that we had discussed with Martin because it was actually fascinating, and I think it's something novel that we have found, but we do see a nice dose-dependent increase in mature C-band, and we do not see as much increase in the activity. So we will expect to see higher levels of activity to the levels of CFTR that we're actually producing. So we are investigating that, and we're also putting in some, implementing some troubleshooting to try to figure out what is this. But it is clearly that the CFTR we're generating is due to the de novo introduced CFTR protein in the lipid nanoparticle. That's for sure.
So thank you very much. With this, I think we have finished this session. So I want to thank all the presenters for their very, very interesting presentations and also the responses. And with this, Megan and myself, we thank you also, all of you who are in the cloud everywhere, for attending this session. Thank you very much, and see you in live, I hope, next year.
Appreciate it. Bye.
Thank you.